wind you up. Well, he's taking it about as graciously as you can. So it's not like they had that incredible play. Dissected it all right.
Yeah. I didn't know that came out.
Here they got this great fun. It's going to be no time out.
He's going to still
They had to make sure the clock stopped.
maintain some aspect.
Hey, they got 30 so I said they had time. They could take time. I appreciate it.
All by their residents.
If they didn't make it,
So we're just having. I'd like to see more.
The only delegation obviously in ours,
exactly. And he may not make it. I don't doubt that. That's the same argument against everybody was there. They were all in a village. So he throws it away. It was actually, I'll tell you what, it's like after that. This whole city, yesterday wore black suits. Anyhow, we got this other 2 years. It goes to Guinea, then into New Caledonia, Great Barrier Reef, Sydney. It's just way off the beaten path.
That's my memory feels like it's working very well.
For a balance.
Are you signed up?
And right.
Yeah.
Just to say, but they were 2 years ahead. We're trying to see our travel again. Sign up that early. It still looks like 500. That's not what I'm sure. We don't. So 10 house. We don't do that.
We've got it. We always went through a year.
Memory feels working as good.
Most of us do 2 other scientists. So we have anywhere from 6 to 8 fellows.
You know where it is? It is basically Western Indonesia.
Well, just the administrative fellowship program. Just has
That's kind of
time.
The info.
The workers are getting rid of them. Boy, that's what I'm like. You got to take your lawyers. You got to always
People in the lab, they work on their smells. Our clinical research, yeah, really exciting. Yeah.
We're all in mourning. I wore a black suit to work yesterday with a black tie and a white shirt. At least twice.
brutal.
That's a really large change.
I guess I don't see it. Maybe a fade.
He was only second down to run a couple of times.
It was a whole.
A lot of scientists are interested in translation.
Well, I haven't remembered when it was back for
Yeah, so that's
There's some weird
Mike White. Mr. White.
yeah. We don't want to
It is true. You do hear that stuff. Is that how people No, you know what? That was what they were coming up with. To get internet service, it's $55 for 2 hours. So, we still use probably $150 $50. Didn't want to use them now. It was costing so that's why I did it.
Regulatory stuff.
I should go back and found that.
Yeah.
This is to, of course, remember what they were
One was journal, because we had to change, got the ID speaker on there, he had to go to Atlanta. He had to go to Atlanta for a full
That's the
disease officer. So we had to move in.
yeah.
I'm not
Match for scientists and educators. We're modeling a lot.
And then and stuff. And then two of the controllers. That's soccer is on some sticks. So it's a story there. This is for yourself. Tell you this is for the good stuff.
Let's go ahead and get started. Continuing our annual Rheumatology for the allergist. We've got a new speaker from our colleagues in Rheumatology, Grant. I don't know why Corvallis. Are you trained Corvallis?
A long time ago.
Well, somehow that came up in CV. So that's the word of the day.
Go Beavs.
So we're going to have an update on rheumatology, I think, primarily on lupus. Thanks for coming.
Yeah. All right, let's get the. Well, I don't know who to thank for inviting me because I was, I think, invited by Greg Gardner. He was actually.
He abdicated.
Yeah, he did. He's been abdicating a lot recently. Which is good. So I'm happy to be here at this unbelievable hour. 7. Is this usual for allergists? It's a little more like 9 o'clock people.
There's a long history how it got.
Yeah.
Okay.
More than you want to hear.
Okay. And I traveled all the way from across the street. My lab is in the East Building. I am in the Division of Rheumatology. I lab, I see patients, and I also am involved in the training program, so I wear multiple hats. But I talked to Greg about this, and instead of going, a whirlwind tour through clinical rheumatology, he said that you all are very interested in sort of updates and hot topics that have relationship to allergy and immunology. So I picked a couple topics that are linked pictorially here by a cell type in the middle, and you probably may already know what cell type that is. Topics that are very interesting, they're hot topics, and they're also areas of focus here in South Lake Union and UW Community. I wanted to highlight a couple of those, two of those topics. I have no disclosures. And the two topics are the involvement of the type interferon pathway and the pathogenesis of lupus. Sort of old questions, new answers type topic. And the second one, I've stolen the title from Jonathan Grack down at UCSF, who shares my interest in this topic, Rheumatology on the Streets, Cocaine-associated Autoimmunity and Rheumatic disease. And there's some significant overlap with some things that you see here as well. And what links these two, the neutrophils and a somewhat recently recognized form of neutrophil activation called NETosis. We'll talk more about that and how these two things are linked. Sort of a little mystery up front. And in case you want to drift off now, I'm going to review key points now and periodically through the rest of the talk. And feel free to interrupt me anytime with questions, and I'll pause in the middle to take some questions as well. So aberrant activation of the type 1 interferon system is pathogenic in SLE, and it contributes to loss of tolerance to nuclear antigens. It's integral into an amplification loop mediated by immune complexes. It's also involved in activation of neutrophils and release of autoantigens, which is sort of a new discovery. And it's also a target of sexual dimorphism in SLE risk, which is my area of particular research interest. And then changing gears, we'll talk about how illicit cocaine use is associated with the development of anti-neutrophil cytoplasmic antibodies, several clinical syndromes, including midline destructive lesions, which most of you are familiar with, but also newly recognized phenomena of thrombotic skin disease, neutropenia, perhaps vasculitis, and talk a little bit about how this drug-induced form of autoimmunity is different than idiopathic pancreavasculitis, and how it links in the same pathway of neutrophil activation, which is now found important. So early in the morning, a little inspiration. My daughter's a soccer player, and we're all big on the World Cup that's coming up here in 2015, just north of the border, Women's World Cup. And this is Shannon Boxx, who's a 3-time Olympic gold medalist, member of the U.S. women's national team, also the Chicago Red Stars professional soccer team, and achieved most of this in spite of her diagnosis with systemic lupus in 2007. She's currently on the squad trying to earn a spot for the World Cup team 2015. So, inspirational story to start off. And she's also a spokesperson for the Lupus Foundation of America. All right, so just a little bit of review on SLE to get everybody up to speed. It is a multi-system autoimmune disease. It's not that common. It's about a fifth or a tenth as common as something like rheumatoid arthritis, and has, as I think most of us know, pretty peculiar demographic in that it primarily, its onset is primarily among young women of reproductive age. It's a 10-to-1 female-to-male ratio, and has a significant genetic component, about 65% inheritance in most cases. And kidney disease is a major source of morbidity and mortality. So the immunologic hallmark of lupus is loss of tolerance against nuclear antigens. And these include chromatin and its components, histones and DNA. And that's actually what gives that homogeneous pattern on anti-nuclear antibody staining. You've got chromatin throughout the nucleus. But also components of the nuclear machinery, including small nuclear ribonuclear proteins, of which Smith antigen and RNP antigens are members, as well as some other nuclear and even some cytoplasmic antigens, Rho and LA, that are part of nuclear machinery as well, particularly processing messenger RNA, probably induction of interfering RNA as well. And these give rise to the characteristics. So it's sort of a targeted autoimmunity. And so why is it a systemic autoimmune disease? Well, I think most of us in the room know that this is a prototypical type 3 hypersensitivity immune disease, where IgG autoantibodies encounter autoantigen, which in the case of lupus is dead and dying cells, components of the nucleus and the nuclear machinery that are formed in circulation. I'm going to talk about this in a little bit later, but the source of those autoantigens has been a big mystery, and it still is, but there's some new clues on where those antigens come from. But these things form circulating immune complexes, and these immune complexes can deposit in various end organs, such as the kidney, and induce local inflammation, tissue damage through complement activation, and binding back, activating FC receptors on myeloid or hematopoietic and non-hematopoietic cells. Lupus also involves type 2 hypersensitivity, so autoimmune hemolytic anemia is a great example of that. But it's probably also involved in other, and for example, nephritis as well. There's good evidence that immune complexes or antibodies bind to antigen exposed, aberrantly exposed in the kidney. It's also probably involved in skin disease, where UV light induces apoptosis of skin cells and releases or exposes autoantigens for binding of autoantibodies. And then type 4 hypersensitivity, which we won't go into much detail, is also activated quite strongly in organs, target organs of lupus. And I won't talk about this much, but there's emerging evidence from mouse models that IgE, immune complexes, and activation of mast cells is involved, and particularly in nephritis, which is fascinating. The type 3 hypersensitivity is the dominant form, immune-mediated lupus. And so the circulating immune complexes, which are formed with antibodies and autoantigens, could be triggered by things that cause cell death, particularly of circulating cells or in tissues, but UV light, bacterial infection, viral infections. But these circulating immune complexes, as I mentioned before, deposit in organs. And here's just an immunofluorescence of a kidney with IgG staining in the skin. Here's the lupus band, IgG in the dermal epidermal interface, et cetera, et cetera. So it's not that lupus targets multiple organs individually. It's that it's a type 3 hypersensitivity disease, and immune complex deposit in a variety of organs. And this is one of the big reasons it's a systemic autoimmune disease. All right, so enough about basics of lupus. I mentioned that the lupus is 66% or so heritability. So that's necessary for lupus in most cases, but not sufficient. Some sort of modulators are required. And we'll talk a little bit about this later, include sex-specific factors, and this contributes to the 10-to-1 female-to-male ratio, environmental triggers, and stochastic events. This is, I think, an increasingly recognized factor in a lot of polygenic diseases, including cancers as well. But it applies to autoimmune disease as well. As Lars Rundblom in Sweden, as a famous lupus researcher said, it's just bad luck. Random events stacking up. So what about the genetic variation? So in 2008 and forward, there's been several genome-wide association studies performed in large lupus cohorts. And what they've identified mostly are common genetic variants that occur relatively low risk of lupus, of 1.1 to 2.0 odds ratios. What they and a few sort of intermediate risk loci, particularly around the class 2 alleles. But these studies don't pick up these rarer genetic variations, such as are listed here, that are picked up more in familial studies. And all the known genetic variants only account for, I think, about 30 to 40% of the heritability. So there's a lot of unidentified risk fields out there, plus the contribution of epistasis, so the shuffle of genes. So certain combinations of genes are worse than others. And I'll come back to this in a few minutes. What's really, I love this picture. It's one of the best diagrams that I've seen related to lupus genetics. But what's clear from these studies is that the risk loci seem to cluster in 3 major pathways. There's one up here on the top that's involved in immune complex process, sort of the clearance and clearance of dead and dying cells, which is a natural process. It goes on continually in our bodies. So think about neutrophils, dominant white cell are in circulation with a turnover time of 12 to 24 hours. Those have to be cleared constantly. But defects in those processes predispose to lupus, as do genetic variation in innate immunity, particularly type 1 interferons, which we'll talk about in much more detail in just a second. And then immune signal transduction here in the lower right, and this really impacts lymphocyte selection, survival, and activation. I think, for the sake of time, I'm going to go over this pretty quickly. This is an idea that I think is not generally appreciated even within the rheumatology community, is that the pathways that lead to loss of tolerance to nuclear antigens are not necessarily the same ones that mediate immune injury. And this has a lot of implications for interpreting GWAS studies, for designing therapies. For example, the genetic variation in T and B cell selection really has very little to do with end organ damage, which involves thrombosis, cytotoxicity, complement activation. There are some pathways, such as Fc receptor signaling, which are involved in both clearance of apoptotic cells, but also immune-mediated injury of immune complexes. So they're involved in both aspects of disease. And as we'll talk about more, type 1 interferon is probably involved in both pathways. And then you have things like complement, where deficiencies in complement actually predispose to risk of lupus. The complement activation is a critical step in the organization. And so I think this is a framework that I think is emerging now and being supported by genetic studies that really will inform better design of therapies and smarter interpretation of genetic studies. I'm not going to touch on this too much. I just wanted to introduce this idea to you all. So, I've been talking about interferons a lot. This is actually the first magazine cover that I remember clearly as I don't remember how old it was, but I was a kid. I just remember this particular cover. I went back and just Googled it, and it was right there, exactly as I remembered it. Where relating to the first recombinant, I think the interferon, I think it was probably gamma gene into E. coli. So it's recombinant production of interferon. There was a lot of excitement over its anti-cancer properties, which, haven't really panned out that much. But this was 1980. Fast forward, 35 years to 2015, and there's been a lot of study in the interferon family. And I just want to go over some nomenclature here. There's 3 known types of interferon in humans, type 1, type 2, type 3, that are grouped actually by the receptors they signal through. So type 1 signal through the type 1 receptor, type 2 interferon gamma through the type 2 receptor, and type 3 through this dimer listed here. Their cellular sources are quite different. The type 1 interferons, particularly interferon beta, are produced by fibroblasts and macrophages, where alpha interferon is produced by cell type plasmacytoid dendritic cells, which I'll talk about in a second. But all of the type 1 interferons seem to be critically involved in antiviral host defense and antiviral immunity, and in some cases anti-protozoal defense. Whereas gamma interferon, as many of us know, is involved in antiviral host defense, but also in macrophage activation and other forms of cell-mediated immunity. And then we won't talk too much about the type 3s. They were only recently described being better characterized as we speak. So, with that background, let's move a little bit more into how type 1 interferons are induced. There's 2 major pathways. One is a cytosolic pathway, and one is an extracellular pathway. And both of these pathways lead to induction of type 1 interferon, plus other cytokines as well, inflammatory cytokines. And these are really interesting. So what these systems basically are doing, these are innate immune recognition systems that sense danger. And the way they do that is through pattern recognition receptors that recognize unique molecular patterns, patterns that are unique to pathogens. So things like double-stranded RNA, which is not a normal intermediate of eukaryotic nucleic acid synthesis, or certain types of hypomethylated CpG DNA, which is found in bacteria. They also recognize the things that induce type 1 interferon are nucleic acids in the wrong place. So the wrong nucleic acids in the wrong place. So cytosol should not be a place where nucleic acids hang out. And if they persist or synthesize in the cytosol, such as with a viral infection, this triggers a variety of cytosolic sensors that then activate production of type 1 interferon, which has antiviral properties, which we won't go into in much detail. There's also an extracellular pathway where cells, particularly myeloid cells, can sense extracellular DNA or immune complexes, endocytose those or phagocytose those, which then triggers pattern recognition receptors such as toll-like receptor 7 and toll-like receptor 9 to induce type 1 interferon as well. Toll-like receptor 7 recognizes viral single-stranded RNA, and toll-like receptor 9 recognizes both viral and bacterial DNA. we'll come back to this in just a few minutes. So back in before 2003, it was known that the blood of patients with lupus had interferon activity. And this is a study from 2003 really showing what's called a heat map of patients. So there's on the y-axis there are a whole bunch of different genes, and their color denotes whether they're up or down based compared to healthy controls, or actually to a baseline. And what you see here in red is up and blue is down. In this middle section of lupus patients, you see clustering of upregulated genes. And then there's also another inflammatory disease control here. And this top cluster are interferon-inducible genes. And there's also a granulopoiesis signature. So this was one of the first hints that the interferon system is activated in lupus patients. And it was also shown that the serum for patients with active lupus had interferon activity, could interfere with viral replication in vitro. And strikingly, you could take the serum from a lupus patient, incubate it with normal blood cells, and induce interferon production. So there was something in the serum of lupus patients that was inducing interferon. And it wasn't just all and it was actually a certain cell type in the blood that was responsible for all of that interferon production. This was work done by Lars Rundblom, Sweden, and others. And it turned out to be something called plasmacytoid DC, pictured here, which is less than 0.5% of the total blood leukocytes, but produces all of the interferon that's induced by lupus serum. And it's a cell type that's now found to be very important in, not surprisingly, antiviral defense against certain viruses. So what is it in the lupus serum that's inducing this? Well, it's immune complexes. They activate plasmacytoid dendritic cells directly through these toll-like receptors that I mentioned that sense RNA and DNA, which is contained in these immune complexes, and also requires internalization via Fc gamma R2A. So this was
those dendritic cells circulating.
Sorry.
Is that normal? Do we all have circulating dendritic cells?
All of us in this room have, if you take our PBMCs, we'll have anywhere between 0.1 and 1% plasmacyte. So they're largely circulating cells that can home to sites of inflammation. In fact, they're present in allergic disease as well. Rhinitis has a pretty big infiltration of cells. And so why is this important? Well, first of all, interferon alpha probably relates to a lot of common symptoms of lupus. So I didn't mention this before, but the degree of interferon alpha in the blood correlates tightly with disease activity. And interferon alpha probably relates to a lot of constitutional symptoms in lupus patients. So all of us here have had a viral infection. We know it's the myalgias, the fatigue, arthralgias, low-grade fever associated with that. Well, active lupus is characterized by those same symptoms. And it's the same symptoms that people get if you give them interferon alpha to treat, for example, hepatitis C virus infection. So the interferon alpha is probably driving a lot of constitutional symptoms in lupus, but it also is acting on the immune system. So I said that interferon alpha drives antiviral immunity. Well, it turns out it has pleiotropic effects on the immune response to help drive, for example, differentiation of T helper cells, differentiation autoantibody or antibody producing B cells. So it acts on a lot of different cell types to really lower thresholds of activation and drive forward adaptive responses. And for these reasons, it's thought to actually amplify lupus autoimmunity in a variety of ways. One of the more important ways is it actually acts back on plasmacytoid DCs to prime them to produce even more interferon alpha. So it's a potent amplification loop of production of antiviral cytokine in the absence of virus infection that drives autoimmunity. We're going to talk about how it acts on neutrophils in just a second. So in this model, we've got autoantibodies forming immune complexes inducing type 1 interferon production by plasmacytoid DCs. Well, was that the chicken or the egg? So where do the autoantibodies come from, and how is type 1 interferon related to that? Well, what we know from mouse models is that the type 1 interferon system is necessary for the development of autoimmunity. So, in other words, in the setting of genetic susceptibility, the type 1 interferon system is critical for the evolution of autoimmunity and subsequent end-organ damage. And so, if you knock out, for example, that type 1 interferon receptor in lupus-prone mice, almost universally across different models, you will ablate or even abolish disease development. So it's necessary for disease. And just recently, just a few months ago, it was shown that if you actually ablate plasmacytoid DCs in lupus mice, you also greatly reduce disease. So both the plasmacytoid DCs and type 1 interferon signaling are really necessary component for disease progression in mice. Well, what about in humans? There's some good evidence from the hepatitis C population that I talked about before. So treatment with interferon alpha, an antiviral cytokine, is therapeutic in patients with that chronic viral infection. Patients treated with interferon alpha have a very high rate of developing subsequent anti-nuclear antibodies, and in some cases clinical rarely lupus, sometimes autoimmune thyroid disease. So we know from that population that treating people with interferon alpha increases or risk of autoimmunity or unmasks late. But is type 1 interferon overproduction sufficient to drive autoimmunity? So then the answer probably is yes, since this comes from very recent discoveries in Mendelian immune disorders, now being bunched together as interferonopathy. So these are diseases of dysregulated type 1 interferon production. And what we're seeing in these diseases is a pretty strong autoimmune phenotype with a lot of features of lupus. So the Aicardi-Goutieres syndrome is probably the best known example. And this is a disease of neonatal or pediatric onset microcephaly, developmental delay, and brain leukodystrophy. And what was recognized early on is both in the blood and in the cerebrospinal fluid, there is an interferon signature very similar to that of the lupus patient. And this starts early in life. And these patients develop anti-nuclear antibodies, anti-double-stranded DNA antibodies, vasculitis. And what's really interesting is that there's all the patients with Aicardi-Goutieres have deficient or genetic mutations in genes that limit the activation of type 1 interferon through that cytoplasmic pathway I mentioned. And this is a nice diagram from our own Dan Stetson, who works across the street here, who's pioneered some basic science in this area, really showing how deficiency in two molecules, TREX1 and or RNase H2, can lead to overproduction of type 1 interferon and autoimmunity. So it turns out that there's a natural sort of tonic level of DNA in our cytosol that comes from endogenous retroviruses and retroelements that are constantly being reverse transcribed. And if those nucleic acids either aren't prevented or they aren't degraded, they are sensed in the cytosol as a viral infection, drive interferon production. So in kids missing TREX1, which is a DNase that normally chews these things up, you get an accumulation of endogenous DNA in the cytosol, which triggers interferon production chronically, tonically producing interferon, which probably leads to a lot of these phenotypes on the right. Or, for example, those who don't have RNAs, that degrades that initial RNA transcript from the retroviral elements that leads to a similar phenotype. All of these molecules listed below are involved in dampening this activation pathway. So this is really new and exciting evidence that aberrant production of type 1 interferon is sufficient to induce lupus-like autoimmunity. So now back to that old question, why do women get lupus more than men? Well, it turns out, and this is the area that I've really been interested in, it turns out that type 1 interferon response is highly sexually divergent. And this is just a pictorial overview of the genetic response to yellow fever vaccine, which is an RNA virus that activates type 1 interferon through that TLR7 pathway, that extrinsic pathway I was talking about. And what you see on the very top is a Venn diagram with genes regulated up in men, women on the left, men on the right, and those that are common between them. And what you see pictorially on the bottom, all the purple genes are upregulated, and the green ones are downregulated. You see a very different, significantly different upregulation of genes in women in response to the yellow fever vaccine than men. And most of these genes cluster with the type 1 response. So this is just an example showing pictorially the vast sex-dependent difference. And so how does that occur? What are the mechanisms? Well, let me back up a little bit. The teleologic reasons behind that we could debate for hours here. And they probably relate to select immunologic requirements of placental reproduction, implantation, and things like that. So, but that's a topic for another day. What are the mechanisms? Well, a lot of work done by other people, work that I wish I would have done, really shows how estrogen is an important driver of type 1 interferon responses. And this is just a diagram from a review Divakar Chovy and I at Cincinnati put together, where estrogen is this little green dot. And there's two places where it works. We think it works to enhance interferon responses through feed-forward mechanisms. One is at the level of the plasmacytoid dendritic cell. There's great evidence now from both humans and mice that estrogen acts on plasmacytoid DCs to enhance interferon response, in part through upregulating adapter molecules downstream of those TLR signals, and also through a feedback mechanism where interferon feeds back on the cell and upregulates estrogen receptor expression. So there's this amplification of estrogen and interferon signaling plasmacytoid disease that seems to occur in parallel in macrophages with interferon beta production as well. And so this appears to be a really important node of interaction between gonadal steroids and autoimmunity, perhaps viral events as well. But even more recent discoveries suggest that hormones aren't the whole picture. There's an experiment that was published last year that I think was, probably 30 years. I'm surprised it didn't happen 30 years ago. But basically, you take lupus mice, right, which we know that have the same sex predilection as humans with lupus, and you just take the marrow out of male mice and put it in female, and you take the marrow out of a female and put it in male, and you see what happens. This is before they reach puberty. So in other words, you're looking at the genetic component to the sex discrepancy. It turns out that a lot of that sex discrepancy in the mouse model of lupus is encoded in the hematopoietic stem cells in their DNA and not dependent on gonadal factors. And what are those factors? Well, I think all of us know that in women, females have two X chromosomes. One of them is inactivated to ensure gene dosage equilibrium between males and females. But what is not as widely appreciated, a certain portion of genes escape inactivation regularly in the X chromosome. A lot of this work is being pioneered by Christine Distache here at UW. And among those genes are genes involved in type 1 response. So it's possible, and this hasn't been proven yet, that women have additional gene dosage of genes involved in the type 1 interferon response. And then this is work that was actually published last year in Nature, and if you're at all interested in the subject, I strongly recommend reading this Nature publication at the bottom. It's pretty short, but really exciting. It turns out that the Y chromosome isn't as useless as it's made made out to be. It actually has Y homologs of X genes, and these are dosage sensitive, which means that you need 2 copies in order to buy 2 functional copies. But it turns out that because they're non-recombining, some of these homologs, they've undergone sex-specific selection in males, and they've actually diverged functionally. So there's functional divergence of these homologs, many of which, or some of which, are involved in the type interferon response. Some of them are involved in broad cellular activities such as ubiquitination, transcription, which introduces this whole idea that cells have sex at the individual cell level, which is totally new idea, which I think is cool. So I need to get through. So what do genetics tell us about the sex difference? Well, here's on the left is the makeup of the average woman who develops lupus, involving genetic, environmental, stochastic, and a large portion of sex-related factors that I just described. Well, what about a man? Man doesn't have the estrogen, as much estrogen, doesn't have that double X genetic component. So how can a man get this? Well, one hypothesis is that a man's got to have more genetic risk. In fact, that's what's being discovered now. So if you look at male versus female lupus, you identify more risk alleles in male lupus than females. And what is the nature of that additional genetic risk? Well, I don't think it's terribly surprising that it involves genes in the type interferon response. DLR7, SPP1 is osteopontin, IRF5 is a signaling molecule. It's the one that I mentioned that's regulated by estrogen. So I don't think this is random. And so in other words, in order for a man to develop lupus, with given equal autosomal genetic risk, for a man to develop lupus, on average, has to have additional genetic risk to compensate for that risk which is conferred by female factors, or to overcome those protective factors in males. So that's really interesting. So I'm going to zip through this. So what's the source of autoantigen in lupus? Well, here's a new source. It turns out the neutrophils can be activated in a form of programmed cell death called neutrophil extracellular trap formation, where they basically spill their DNA, their chromatin out. It's laced with antimicrobial granule proteins, and is thought perhaps to trap bacteria and help neutralize them, although that hasn't been proven yet. Here's a picture of one. So in green, you see H3 histone protein. Blue is DNA. So you've got 2 healthy neutrophils on either side. In the middle neutrophil that's the net, extruded its chromatin. There are multiple triggers for this, including inflammatory cytokines, bacterial products. But as you've probably guessed, you've just dumped into, if this happens in circulation, which it does, you've just dumped into circulation a bunch of autoantigens recognized by autoantibodies. So there's a lot of excitement about this being a potential major source of circulating autoantigen lupus. And what do these immune complexes do? Well, they induce interferons. It turns out that you take nets, the DNA from nets, incubate with the lupus autoantibodies, they trigger plasmacytoid DCs to make interferon alpha, just like immune complexes for lupus patients. And importantly, that interferon alpha acts on the neutrophils to prime them to undergo this netosis process. And those immune complexes actually act on the neutrophils as well to prime them. So it's another identification of another feed-forward amplification group driven by interferon alpha, potentially providing a source of autoantigen in circulation in these patients. So early days on. So here's a summary, and this is in the handout. I'm going to not go over that, just for sake of time, so we'll get to our final, more clinically relevant topic.
Just before you move on, is that story true broadly of autoimmune diseases like RA and others that you would categorize?
So, great question. So there's intense interest on the involvement of these nets in RA and ANCA-vasculitis as well. And it turns out that they are important, but in very different ways. So it turns out that this netosis, the histones and antigens that are released are modified. They're actually citrulinated, so they're modified. A special modification that's actually specifically recognized by autoantibodies in RA patients. And then it turns out that these nets can be deposited in end organs where they induce thrombosis and local inflammatory responses that could be involved in ANCA vasculitis. So this type 1 interferon induction pathway seems to be relatively specific to lupus. Yeah.
Do these patients have any defect in their caspase system to not get rid of these DNA particles when they're released?
That I don't know, but I do know that genetic variations in DNAs, so extracellular DNAs, is a major risk factor for lupus. So an inability to degrade these nets, which are normally degraded very quickly in circulation. Yeah.
Well, on that topic, so what normally degrades them? How is it clear?
So DNases in particular, and they probably are cleared in other end organs, perhaps the spleen, by macrophages, although it's not really clear. And it's not clear to what extent intravascular netosis occurs. We know it occurs, we just don't know how robust it is. So I'm going to change gears here and talk about a case. This is somebody you might see in clinic, actually. Somebody with recurrent epistaxis, frontal pain, sinusitis, has a history of hypertension, and some mild chronic kidney disease, no other medical problems. And rhinoscopy reveals this septal perforation, crusting, and probably super infection. You're worried about limited Wegener's granulomatosis, among other things. You get ANCA testing, you see high titer ANCA, but sort of a confusing pattern of autoantibody. So which of the following is most likely to be a diagnosis? So I think I've given away the answer in the yeah. Talk about why in a second. So I just want to highlight some new things in the rheumatic manifestations of cocaine use. Talk about these three entities and the relationship to anti-neutrophil cytoplasmic antibodies. So here's cocaine. Comes from the erythroxylum coca plant, which is native to the Andes in South America. It's probably used for centuries as a stimulant, aphrodisiac, and in Western history was used as an over-the-counter analgesic, and in more recent decades as a drug use, epidemic of use. This is cocaine. Just want to review the clinical features of cocaine-induced midline destructive lesions, usually associated with snorted cocaine. Patients present these features. And chronic sinusitis is quite common, and it can look very much like limited Wegener's. I think if you showed these pictures to rheumatologists, allergists, and ENT docs, they probably couldn't tell you about history, what it was like, almost looking at. Although these are mucosal isolated diseases, systemic manifestations, and they're almost always so let's get into that. A little bit on the differential of CIMDL. So includes the ANCA vasculitides, particularly Wegener's, which, as I hinted at before, should have systemic involvement. Sarcoidosis always has to be considered. Generally, these patients have extra mucosal manifestations, as listed here. Infections to consider. I work at Harborview, so TB is on every differential diagnosis we have. And then the diabetic, we need to think of mucormycosis. Syphilis is also possible. I don't think any of this is a surprise to you here. Oftentimes you'll get a biopsy in these patients, but the specific, the diagnostic yield of these biopsies is quite poor, differentiating these diseases. So the assumption is you're not going to get a diagnosis with biopsy, but if you do see something that is is specific, it can help you differentiate these syndromes. And in particular, in CIMDL, you're going to get a mixed inflammatory infiltrate, but you shouldn't see vasculitis, you shouldn't see granulomas, and you shouldn't see predominantly eosinophilic. Whereas in GPA, which is Wegener's, you'll see the same mixed inflammatory infiltrate, and if you do find specific findings, you'll see vasculitis and granulomas and giant cells. So in other words, if you see the granulomas and giant cells, it's probably not just cocaine. You're probably dealing with either a limited form or an initial manifestation of systemic vasculitis. And if those findings are associated with predominant eosinophilia, you might be dealing with our friends Churg Strauss or Egba. So here's just a picture on the right of sinus biopsy from somebody with Wegener's granulomatosis. And B is actually bone fragments, stars are granulomas, and triangles show giant cells. And the ovals are just mixed. Okay. So, what you learned in med school. So, I went back. I wasn't quite sure when ANCA was first described, but it was back in the mid-'80s, early to mid-'80s by Davies and others. So, may or may not have learned about this in medical school. But the two autoantibodies associated with idiopathic vasculitis are these listed, and they're what give rise to these immunofluorescence patterns. So the anti-proteinase 3 gives rise to the cytoplasmic ANCA stain in neutrophils and oxidase. This is actually an artifact fixation process. Doesn't reflect their actual cytoplasmic localization in vivo. It's an artifact of form and then ethyl fixation. So what about the ANCA in our cocaine-induced disease? Well, it's generally high titer p-ANCA, but generally negative anti-MPO and low-level PR3. So there's unmeasured autoantibodies. And what are those autoantibodies? It turns out they're disease. And this is a nice study from 2004, really showing, this is a log scale on the left showing serum anti-neutrophil elastase antibodies in CIMDL patients compared to microscopic polyangiitis, controls, and various other autoimmune diseases. So I want to come back to this in a second.
Did you measure that then if you're worried about?
So it's not clinically available in the UW system. There's some labs you can send it out to. So we're actually working towards adding it to the UW system lab for just this reason. So in the last 10 minutes, I want to really talk about this exciting syndrome that we're encountering. This is the first case we encountered in 2008. 46-year-old woman with chronic hepatitis C was transferred from purpura in the legs and abdomen. And this isn't her, but it looked quite a bit like this. Very well demarcated areas of blistering and purpura. Quite devastating. This patient was a user of crack cocaine and amphetamines. She also had severe neutropenia. And we were initially concerned about coagulopathies, vasculitis, or precipitate disease. All of that workup was negative. In her vasculitis workup, she had a low-level ANA. She had normal complement levels. She had a p-ANCA level greater than 1 to 65,000, but relatively low MPO, PR3, and double positivity, which I didn't mention is quite unusual for the idiopathic vasculitis. Skin biopsy showed diffuse fibrous clots in the dermal vessels, but no vasculitis. And this is a picture here showing some of these fibrin clots in the vessel and some infiltrate. It's all perivascular. There's no vascular wall destruction. So no vasculitis. Since then, we've seen dozens of these, maybe almost 24 cases similar to it been reported across the country. And they coincide DEA reports of contamination of street cocaine with something called levamisole. And the reasons it's put in are unclear. There's a is it Seattle Weekly? One of the daily or weekly newspapers had a whole expose on the reasons in South America, interviewed all sorts of nefarious types down there. But the reasons are unclear. So, but it's been a significant contaminant of cocaine since 2009 at least. It's an old immunomodulatory drug that was used in RA. And the product syndrome was taken off the market due to toxicity, neutropenia, product conditions, exactly what we saw in our patient subsequent and associated induction of ANCA autoimmunity. And so since then, we and others have described a syndrome that we're calling cocaine-levamisole associated autoimmunity, characterized by retiform purpura, neutropenia. It occurs with active cocaine use and usually abates with cessation, which is, I think, very important clue as to pathogenic. Can occur interestingly with almost any route of administration, so snorted, smoked, IV injection. And these patients present with exceptionally high ANCA titers that are almost always P-ANCA pattern, but with very little MPO, or variably present MPO, and PR3. So there's this mismatch between the immunofluorescence and the measured autoantibodies. There's a general absence of vasculitis, although there's some scattered reports of vasculitic findings such as posse, GN, pulmonary hemorrhage. And these patients are generally treated with supportive care and obvious absence of cocaine. And there's the absence of these vasculitic findings. There's no role for immunosuppression. This is a case series that we presented at the ACR meeting in 2014. Summarized here. These are patients that referred to for rheumatology evaluation who had high-level ANCAs and were cocaine. So there's a clear bias in our selection. But what's interesting is it's almost exclusively female between the ages of 35 and 50. And others, particularly Jonathan Graff at UCSF, reported very similar demographic. So this appears to be very different than the general cocaine-using population. And clinical manifestations are actually more varied than initially than had been reported in literature, including more pulmonary hemorrhage and one case of blottitis, somebody with crack cocaine smoke. And also surprising absence of ANA reactivity and an almost near universal presence of anticardial GM, which is best. So just others have actually looked at immune reactivity in these patients, and you can see in this bottom table that they're universally positive. This is a series of 6 patients. They're universally positive for elastic antibodies, variably positive for PR3 and MPO, but also have reactivity against other neutrophil granules, such as cathepsin G, lactoferrin, but not all. So lysozyme reactivity is not very important. And I just want to highlight that this reactivity of ANCA reactivity against neutrophil elastase seems to be common to all drug-induced ANCA syndromes. So hydralazine and propylthiouracil, drug-induced thyroid disease, can induce ANCA antibodies that are targeted against neutrophil elastase, which is very different than idiopathic ANCA vasculitis, which does not appear to target neutrophil elastase. I don't think I'll go into much detail clinically comparing types 1.
That slide reminds me of drug-induced lupus, and back to the first part of the talk. Mm-hmm. Any of the mechanisms you were talking about relevant to drug-induced lupus?
Well, possibly. And if you look at the hydralazine column, see they had an ANA. So something about hydralazine induces both ANCA and ANA. And hydralazine is one of these drugs that alters methylation and modification of histones. And so it's possible that drugs could alter the immunogenicity of NETs, for example, and make them induce autoantibodies. There's actually some data with hydralazine in mouse that shows just that. You treat mice with hydralazine, and the NETs they make are much more alterations, histones that are associated, they're resistant to DNAs and all sorts of things. So yeah, they're very well could be. And so I think some of you may have already put together this connection. The ANCA in cocaine-associated autoimmunity seems to target major components of NETs, and this is just an enzymatic breakdown of normal human NETs. It's almost all not dominated by neutrophil elastase activity as well as cathepsin G. These are the chief targets of autoantibodies in cocaine-induced autoimmunity. Also, netosis is a form of neutrophil cell death. It could account for neutropenia in these patients. Intravascular NETs, as I mentioned earlier, cause thrombosis, and this could be disease. And so, collaboration with the head of the division of Rheumatology, Keith Elkin, and a postdoc, Christian Reed there, we've really been studying what cocaine and levamisole do to neutrophils in vitro. And indeed, they can individually induce NETosis. So it's just a picture of normal neutrophils without stimulation with cocaine at a physiologic dose after, this is 12 hours, and this is levamisole. This is PMA, this is a quantification of the right chemical inducer control. And so it's possible that cocaine and levamisole are involved in activating neutrophils and releasing autoantigens in these patients. And so we're actively studying this question. There's a whole bunch more questions that I think are really exciting. It's like, why does it appear to occur in subsets or demographics across our system to see if it's really true? Could this be a very common form of drug-induced autoimmunity that's going unrecognized? We don't know what the ANCA positive general cocaine population is. We're going to find out. Harvard is a great place to do that. What role does ANCA play in this disease? As I mentioned before, these patients are healthy unless they get cocaine. Then they get a bout of disease, and it goes away if they stop. So something about cocaine is maybe releasing autoantigens and the presence of autoantibodies that could spur disease. And how does cocaine and levamisole appear to break lungs? We're very interested. It turns out that the neutrophils that cocaine and levamisole are activating also are cellular reservoirs of a molecule called BAF, B-cell activating factor, which is really important, appears to be important for breaking tolerance in lupus models. Could it be that this is involved in initial tolerance? So these are all really interesting questions of a newly recognized form of drug-induced autoimmunity that you might see. And so to summarize this section, illicit cocaine use is associated with anti-neutrophil cytoplasmic antibodies and a variety of clinical syndromes, including isolated upper airway disease. Clinically, if you should suspect a drug-induced ANCA when you see a very high ANCA immunofluorescence, particularly P-ANCA, that doesn't match with PR3 and PO reactivity. Importantly, I think the pathogenesis of these syndromes could very well likely involve drug-induced neutrophil NETosis. So I want to finish with a bright picture to get your morning started.
That was very good. In that slide you showed that Christian had done, he looked at stimulating with both together because
great
question, are they synergistic? Or?
Yeah, so we did that, and we did not find an in vitro synergistic interaction. In fact, there was some sort of competition going on, which I'm just trying to figure out. That's a great question.
Question on your first part on the interferon alpha regulation. Have people done like microRNA, microarrays? Is there any dysregulation there? Is potentially that could be a target, a therapeutic target, or?
Yeah, and unfortunately, this is not an area of my expertise. I know that there's multiple microRNAs that are dysregulated in lupus, but I'm not aware of any of them being involved in the type 1 interferon pathway. I bet they are, but I don't know that literature well. Yeah.
If you look at the people that were treated for Hep C with interferon, have you seen a giant pulse of these people ending up with these situations down the road?
No. So the relationship between interferon alpha therapy for Hep C and autoimmunity is not that strong. I think, and what you often see is asymptomatic autoimmunity. So you'll see high titer ANA and no disease. And I'm not aware of any long-term outcomes in those patients, but I haven't been seeing them in clinic, 3 years after interferon alpha therapy. So I think that the interferon alpha, as I mentioned before, in the absence of genetic susceptibility, at least low-level, short-term interferon alpha, may not be sufficient to induce autoimmunity. You need either that plus genetic susceptibility or early, high-level, chronic type 1 interferon induction like in the Icardi-Gutierrez patients. But that's just a guess.
And you had said there was a thought that the Ig activation of mast cells might be involved in the nephritis. Is any case reports of people on omalizumab or anti-Ig therapy where
No,
having a reduction?
but I
if
think
they
it's going
could-
to be tried, because there's been reports of mast cells in human lupus nephritis and biopsies, but the study that I mentioned was in 2 mouse models, looking at knocking out the IgE locus and its effect on nephritis. But it does introduce the possibility of using that in human disease. But I don't know the data that well in humans.
Yeah, in a system like the NETosis that you showed with that self-propagating, there's got to be something else that regulates that system to shut that off. Has anybody looked at like T regulatory cells? They're like in their role.
I'm not aware of any papers looking at how. Well, wait a minute. There's one actually suggesting Tregs do control not the NETosis itself, but
like the
end organ damage that can be caused by it. But I think one of the major breaks on NETosis appears to be DNase activity. This may be one reason why loss of DNase is such a strong risk factor, rare but strong risk factor in lupus. And also one reason that people are targeting using DNases and RNases to basically neutralize immune complexes in lupus. So this is a new strategy that's in phase 1 trials. And it might be really helpful in other diseases of NETosis, such as vasculitis.
Do you routinely screen all your vasculitis patients with ANCAs now?
Well, it's part of the workup for somebody in whom you suspect has systemic vasculitis. So as I mentioned, the case series that I talked about has a bias towards people with vasculitic-like symptoms. Otherwise, no one would have checked ANCA. But I'm really interested in looking at, for example, all cocaine users and looking what the ANCA positivity is and what the reactivity is.
I'm thinking of the more sensitive end of that. If we see a vasculitis patient in our office with a p-ANCA of 75,000, I mean, do we assume that they're cocaine users, or do
I would definitely take, if you
take a better drug history at that point?
Well, yeah, or more forceful drug history, because often we've already taken it, but really, so you suspect drugs, not just cocaine, because you can see that in somebody on hydralazine. So your CHF patient is on hydralazine for afterload reduction. PTU is not used very often, so hydralazine is the one we're going to see most frequently. Yeah, I would suspect that you see a super high p-ANCA titer and it doesn't match the PR3 and MPO. You should have a high degree of suspicion for a drug-induced autoimmunity.
Change this treatment. Get them off it.
Well, thanks very much.
Yeah, you're welcome.
This is the passing of the gathering. Greg will never be back. That's the plan.
I went to the Western Allergy meeting. They got you all talks here.
Oh, he's teaching.
Had a good lineup.
He's focusing on his clinical teaching.
Also, we've got, I think you've already done it, but the ECGME sends out their annual questionnaire.
Yeah.
Have you seen that come through on your...
Have you
already done it? Yet, or- It's just important that this is an important part of the reaccreditation. So if there's any things that you're unsure of, just check with us, or if there's any concerns, talk to Mary's committee to hear things, because it's not a place to vent a problem, because
Sure. We get it.
it can be, you can get a site visit, and in 2 weeks, some questions came up with one student you gave residential lab. It's a crazy sort of situation. So, you already did it, Dan?
I already did it.
Okay.
Drew talked.
But we do need all of you to respond.
Yeah.
So, just check with Drew, me here anyway, before, if you have any questions,
It's the DBR president. It's really weird because it actually doesn't really apply to us. They actually ask, do subspecialty fellows interfere with your learning? And
Yeah,
it's all resident. As a resident, are you in your first year of residency, or are you in your last year of residency?
well, they call it
Something general, so it's very hard and kind of
Thank you. Yeah.
Well, I know everybody enjoyed it.
Matt,
Yeah.
I'll get into that.
Yeah, if you want.
I've been thinking about it.
Yeah. Can you hear?
I'm not making it flow better.
No. Oh, you parked underneath, right?
I parked in the
Did you?
under the East building. Because I actually don't, even though I work here, I never drive.
Yeah. Oh, no worries. Yeah, no worries at all.
I
Okay.
take the shuttle, but the shuttle doesn't run. Yeah.
Okay.
Andy
Yeah,
and
no
Troy
problem.
gave me a little
Yes. So
training.
just let's do this. Okay. And then
So,
what
yeah, I
you
think
want to
probably
do is before you go down to your car, then just check at the front desk with security, and they'll just
They run into
sign you in, or they do their mojo.
I don't.
So.
I take the bus every day, so I don't even drive. I don't have
Oh, that's smart.
for that here.
Yeah, that's really smart. So anyway,
Headed back to the U.
well, take some fruit or something to go there. Okay.
Thanks.
Yeah.
Thanks so much.
Thanks a lot. That was great.
Well, we're trying to reopen the study. And we're trying to finish up our pediatric communication. So, but beyond omalizumab. Oh, yeah. Oh, God. Yeah. We've got an anti-13 coming out, and that kind of thing.